In this video, we will discuss about the chemokines and cytokines that are released after CAR T cell therapies. So here, what the researchers has presented the results for the chemokines and cytokines that are being analyzed after 1 month of the treatment with this CAR T cell therapies, or we can say that after tumor removals with CAR T cell therapies, and compare it with the mock T cells that does not have antigens that can target the tumor cells. So first one is this DIPG, and with this GD2 CAR T cells, and compare it with this DIPG plus mock T cells, and this is for this intrinsic potent glioma cells. And here what they have find out, the cytokines are upregulated when the on-target CAR T cell therapies is used, or when GD2 CAR T cells are used compared to the mock T cells as a control. Cytokines that are been observed are presented here are CXCL10, CCL2, CCL7, CCL11, and the other cytokines are also shown here at its comparison levels with respect to the mock T cells. And for this acute lymphoblastic leukemia, also they used the CD19 CAR T cells and compare it with these mock T cells. And here also we can see that all the four major cytokines are upregulated when in the case where the CD19 CAR T cells on target therapeutic approach is used compared to the mock T cells control approach. We can see that the upregulation is statistically significant. And similar results they have observed for this aggressive osteocarcinoma. In this case, they also use CD19 CAR T cells and compare it with mock T cells. And here we see that most of the cells are upregulated, or at most of the cytokines are upregulated or at same levels compared to the mock cells. And the upregulations here also about the cytokines CXCL10, CCL2, CCL7, and CCL11. These three are statistically significant, whereas for the CCL7 the increase is non-significant. The interesting results observed for this rapid clearing osteocarcinoma, in this case they do not see any significant changes, not just for these four cytokines that are mentioned here, but also for the other cytokines that are been plotted in the heat map. They do not see that when they used on target therapy with this GD2 CAR T cells and compare it with this mock T cells, they do not see an increase in the cytokines. And this is also in line with the results that we have discussed yesterday when we are discussing this NORT and T-maze test, that it is non-significant for this rapid clearing osteocarcinoma. However, for the murine melanoma, we can see the cytokines are statistically significant when they used on target CD19 CAR T cells, compare it with the mock T cells for the CXL10, CCL7, and CCL11, but the CCL2 is non-significant. And when we see its results that we have discussed in the previous video, that here this NORA test is and the chemotherapies for this NORT test and the chemotherapies is, we can see that it's statistically significant, and for the T-maze test also it's statistically significant, but here we do not see significant changes in the CCL2, but in the other cytokines we can see significant changes. And so this is also panel E, this murine melanoma results also in line with the previous results. And when we see the heat map, we can see that most of them are upregulated by factor of 1, and except some of them are upregulated significantly higher, that is CCL7 or MCP3. So these results are also in line with the this cytokines, this cognitive impairment that they have observed using two tests, that is TMAS test and novel object recognition test. So this further substantiates their points that they point that CAR T cells has some cognitive impairment, which is validated by the these two tests that we have discussed yesterday, and also by the cytokines that are upregulated in the CAR T cells. So this is not, you cannot, researchers cannot say that it's 100% true, but in most of the cases it seems like CAR T cells has some cognitive impairment after in a long term effect, as a long term effect. If you want to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.